So I'm Hideyosaki, and thank you for joining tonight. 68th metaPhorest seminar, and today's guest is Ewen Chardronnet and Maria Szukała, and from France and Switzerland, and they're artists and researchers, and who are studying on recently about the possible communication or symbiosis between the humans and photosynthetic organisms. And they are visiting Japan, and will do some artistic research on the Japanese seaweed cultures and the filming, and so and it's great honor to us to have them and on the metaPhorest seminar today. And actually, they visited our lab today and had a very interesting conversation, and we look forward to hearing their background and prospect of their artistic project, that we can learn a lot, hopefully. Yeah. And George, please.
Yeah, okay, thank you, Hidi. You said everything as well. So I'm also very happy to welcome Ewen and Maya here. So I'm also a very proud member of the metaPhorest research group here at Waseda University. It's really the 68th edition of this online symposium. And I think we're gonna take 2 or 3 more minutes to start until everybody's here. In the meantime, I would also point out, so this is part 1 of our lecture and workshops with Ewen and Maya. We're gonna have another round of lectures on the May 31st at BioClub in Shibuya. And at that time, we're also gonna do a speculative cooking workshop. So please come to this as well. We're gonna start at 6 o'clock with the lecture, and at 7 o'clock with the cooking workshop. And I'll also send out the infos on the metaPhorest channel as well. So please join this as well. Yeah. So what are you gonna cook, Maya? Do you know yet? Algae.
I think you're gonna cook. So it's a participant.
Yeah, no, we're all gonna cook. We're all gonna cook together.
The idea is to create new recipes with algae. So
yeah,
it's really about speculating and invent new kitchen gestures as well.
Okay, this sounds really great. I can't wait for this. Okay, so we are 16 now. I think we can start, right? So yeah, as Hidi already said, so Maya and Ewen, they're in Japan now for 2-3 weeks on their research trip, and yeah, I think there's nothing more for me left than to welcome them and hand over the screen to you guys. So please, and I think if there are any questions, please write them in the chat, and we're gonna have a Q&A at the end of it. Yeah. So thank you. 日本語も大丈夫です。日本語の質問あれば、日本語で書いてください。はい、ありがとうございます。じゃあどうぞ。Please start.
Yes, very, very happy to be here in Japan, especially. Also, thank you so much for the invitation. It was thanks to Georg and Hideo that we actually could travel to Japan. Japan is still closed for touristic reason to enter. So we really feel privileged to continue our research here in the country. As Hideo said, I'm from Switzerland. Zurich is my base where I work. I'm also a founder of a BioLab together with Hecteria. I'm co-partner of Marc Dussé. Hecteria is a society that exists more than 10 years, which does tinkering, workshops, and DIY bio around life sciences in general. And now coming here to Japan is also together with Ewen Chardronnet. So this fusion between the Makery and the Hecteria networks. Yes, I'm a chef, artist, and fermentista. So I started with fermentation as a practice. Got really deep into the material, also sourcing the internet on all kind of information on microbiomes, articles, scientific articles, and now I started to do this research on the algae since 1 year, and the final goal is to produce a movie. Yeah.
Hello, I'm Ewen Chardronnet. I want to thank also Ideo and Georg, because as Maya said, we needed professional reason to come here, and that was really nice from metaPhorest and BioClub to have us as guests. I guess in a week we can tell more. We just arrived 2 days ago, and in a week we're going to tell more about our trip. We're going a week in research in the south of the country in the Ariake Bay with Toru Oyama and Cherise Fong accompanying us. So we also thank them so much for helping us in the field research. So on 31st, I guess you hear more about our trip in the south. Today I will. I'm Ewen Chardronnet. As as a day job, I run a magazine called Makery.info. It's not only a magazine, we call it, let's say, a media lab, because we also produce events and conferences and artist residencies, including a festival called Open Source Body, which is about art, health, and biomedical research, open science. And the next edition is in September in Paris. We have a big exhibition this year, since we are also leading a Creative Europe cooperation program with several countries. I see Kira O'Reilly here from Helsinki. We collaborate with BioArt Society, for instance, from Finland. And I also, so I had artistic practice mostly in collective. I like to define myself as an artist collective. I only work in collective practice. And among them, I've been working a lot with Bureau d'Etudes, and I'm gonna talk about you a bit of that, and as well as art and marine biology platform. I set it up with a French scientist colleague from the Roscoff Marine Station in west of France. It's on the Atlantic coast. It's the CNRS National Research Center on marine biology, one of the biggest in France, as well as a very important facility in Europe, hosting a lot of master students and PhDs and a lot of guests from all over the world. And with my friend, we started an artist residency, and I'm going to share my screen now to show you everything. There's a window. Yeah, no. Sorry, 2 seconds. Back to. Yes, here we are. So do you see my screen well?
Yeah.
Super.
We also see your peripheralia. You see the surrounding.
Oh. Maybe I have to.
I think if you click the share in screen, like écran, then it works.
Yeah. Is it better now? Or?
Yeah.
Yes. Yeah, it's not good for me. Right?
Here it is. Look, here it is.
Oh, yeah. And now?
It's the same. It's good.
Is it good, Georg? Yes?
Yes,
You
looks
have
good.
full screen? Yeah, okay. So I will start this story because this lab we created back in 2015-16, and back in those years it was actually the COP21 in Paris, the climate conference in November, December 2015, and I was collaborating for many years with a graphic designers and artist group called the Bureau d'Etudes. They do a lot of maps, maybe you heard about them. And we run this, let's say, artist-run journal called the Laboratory Planet. We make one every few years. It's not a regular publication, but it's for us a tool for doing research. And it's the journal was created in 2007 for the first time at the occasion of Rixi Festival in Riga, in Latvia. So we work on philosophy of science, and we publish it in several languages. The journal format was important for us because it's a way to reach out a wider audience. We printed many copies, like 10,000, 20,000 of each language. So at first we published in English and French, and the last issue was also published in Spanish. In this 5th issue, we decided to approach the debate on, on dating the, back 2015 was this debate on dating the beginning of an Anthropocene age. And we wanted to approach it from a, let's say, xenopolitical angle, since the impossibility of defining a unity of the terrestrial human project in regard of its impact on its primary environment seemed to us to require in-depth reflections on the real agent of transformation, of this transformation. So we like to start with the hypothesis. So our hypothesis was then to attempt to apply this alternative analysis framework and to envision the probability that this major transformation could be the result of a xenopower, destroying the ontologies established by present and past terrestrial humanities. So the first speculative hypothesis being that capitalism could be of extraterrestrial origin. So this provocative first page, this alien capitalism, the world is becoming alien. We want to tackle the xenopolitics of the Anthropocene. I'm not going to go, you can find it online on laboratoryplanet.org. We invited many interesting writers to collaborate. Among them, we published and translated in French and Spanish the Toulousain Manifesto of Santa Cruz, was the first, let's say, version of the text of the Larvae book, Staying with the Trouble. It was a short manifesto. And from the exchanges we got with the Larvae and Vinciane Després to translate, the journal we came to recognize as a recent understanding of human ecosystem and of human impact on the biosphere left to link between the notion of sustainability and co-evolution and harmonization of cultural and biological evolution. Since then, many artists have been tackling this topic. I'm not now being so surprising for many of us, but here it was back in 2015. Following the publication of the Laboratory Planet, I'm from Roscoff. I'm originally from there. My family lives near the marine station. And so it's been a bit of my playground in my childhood to go there, to jump off the wall and go in the lab even as a child. And at some point, a new researcher came in called Dr. Xavier Bailly. He starting to research there, and we became friends in a way because he started to talk to me about his passion and started the multicellular marine models lab. His lab called M3, multicellular marine models. And on the marine station is also a department of the Sorbonne University. And together, he was very open to transdisciplinary collaboration. So working with artists and having a bioart practice, but also with people from, let's say, Fab Lab of BioLab or citizen science as helpers in his research. Sometimes he needs to, design devices, work with Arduinos. So we also collaborated on that level, bringing him some, the marine station is a bit isolated on the seaside and it doesn't have connection to big cities very close. So we organized a bit this collaboration that way. That was artists, but also tinkerers, bricoleurs, or people from the Fab Lab of Nantes or from Paris as well. So we wanted to support artists in the field of marine biology, whether hosting them in residency, but also accompanying them and helping them doing the field research. So here you see some few photos of the different artists who visited us. And for the last 5 years, we hosted Spela Petric and Robertina Sebianic, artist from Slovenia, Carol Thibault, Vincent Poublar, artist from France, Mia Turcic, also from Slovenia, and Nicolas Floch and Rennes, the art school of Rennes in West France, who they have an artist boat hosting residency on the boat itself. So we hosted them then. And so other artists, Jean-Philippe Blanchard, Frédéric Aitou-Haiti, Olivier Morvan, Maya Minder, who is with me now, and Sandra Buller as well. So the M3 lab has a strong focus on animal-alga symbiosis, meaning animals who host algae in their epidermis and get nutrients through their photosynthesis, such as the Elysia chlorotica slug, which you see on the top left, and but above all, the Simsagitifera roscofensis, a 3-4 millimeter long worm, you can see also below, that entirely rely on the algae photosynthesis, and who was described for the first time in Roscoff. That's why he carries this name, Roscofensis, but you can find it on the coast of from Portugal to Wales, and you have probably species similar in the Pacific. As a matter of course, since we were discussing animal alga or animalga as a new word, we decided to invite first the Slovenian artists Spela Petric and Robertina Sebianic, who were conducted the project called Humalga towards a human spore. Just in the years it was we started this residency back in 2012, 13, 14. In this residency context, Robertina Sebianic initiated the first chapter as well of a project called Aquatocene about subaquatic sounds and sound pollution. And Spela Petric started to work on human hormone influence on animals and plants development. And that led her to do then a lot of projects on this topic, and you may have heard about it. In Roscoff, she conducted a study on the impact of human hormones on sea urchins larvae development. I can show you a small video about it and comment it. So she did various control sample, and so you see the sea urchin larvae has a very angular shape, and with different level of hormones, whether progesterone or estrogens, you can see they have modification, and with a lot of progesterone, they become very round and very affected by the hormones. You have testosterone. Compared to the control, you see the larvae get really. Here, estrogen 0.01 micromole, but then with a higher level, it becomes completely round. So that's research she was conducting during residency in Roscoff, as well as working, of course, in the animals alga symbiosis also with us. So a part of the residency and the art project initiated during those times in Roscoff Marine Station, we started following and supporting the scientific work per se of the multicellular marine models lab on the Roscoff worm. And a major thread of the initiative is the design of research protocols involving the photosymbiotic worm, so the Roscoffensis, as a potential biological model to apply to space research. The space mission, so-called now, Roscosmoe, we call it, connects marine biology, space ecology, design, and anthropology of science and intermedia art as well. These photosynthetic marine animals live in colonies up to 7 million individuals on the tidal zone, just when the sea just go away at the very beginning of the tidal zone from so on, already on the beach. And when you look at it on the beach, you see these green lines, and at low tide, and you might think it's the actual algae decomposing, but it is actually colonies of millions of very tiny animals. And the Roscoffensis species is also an iconic species often mentioned by Lynn Margulis in several books of hers, and book Symbiotic Planet starts by describing it as a unique example of symbiosis, because it's an animal don't not eat at all, but completely incorporate the algae in its tissues. And while in Microcosmos book, she gets inspired by the Roscofensis to open up to, let's say, becoming Homo Photosyntheticus of humankind, a path towards an autotroph human with no need to eat anymore, and in the purpose of, you know, space exploration, how we're going to live in space when we can't bring with us a full, you know, higher plants and our food usual regimes. I have to say this really has largely contributed to trigger our interest as artists, this very speculative ground introduced by Lynn Margulis. So the Roscofensis is the vernacular name is Roscoe's worm. Otherwise, you know, we say Roscoe worm. Even in England, they say mint sauce worm. And so it's a flatworm. They can, as I said, you find it from Portugal to Wales, and the origin of and nature of the green color has stimulated very early on the zoologists. So in the 19th century already, in the context of let's say Lamarckian or neo-Lamarckian research of symbiosis studying lichens, but also this species. So the animal hosts a green microalgae, a specific species called Tetraselmis convolutae, under its epidermis. And it was described as a photosymbiosis at the time. And biological investigation on commensalism means the animal host, I mean, embeds the algae, but the algae is not exactly free. It loses its flagella and is a bit captive. So it's not exactly you know, shared symbiosis. It's more like, it's not damaged, but we call it commensalism. And at the Roscoff Marine Station, the British biologist Patrick Geddes questioned himself on the nature of the origin of the green compound of the worm, at the time different name, Convoluta chulci. It succinctly described it chlorophyll containing cells. And in 1886, Yves Delage, who was also the director of the Roscoff Marine Station, published a detailed historical study describing the nervous system and the sense organ of the same worm. And is realized, like, maybe this nature of Zoochlorella, are they really algae? Where do they come from? Was a symbiotic relationship that unites them? And it came with, in 1891, Ludwig von Graf, German zoologist from the University of Graz in Austria, that took the taxonomy, the redescription of the worm in Roscoff Marine Station as well. So he made difference with other species, especially the Schulze you find in Adriatic Sea, and rename it as a Roscoff worm. In the academic book Untitled Plant Animals, published in 1912, Frederick Keibel succeeded to describe the nature and the origin of the microalgae, very interesting book to get, and the specific relation between the Roscofensis and its photosynthetic partner. So that's about the history, but in the lab, our colleague, Dr. Xavier Bailly, developed an educational kit for the national education system in France to understand whether the biology and the symbiosis of the Roscofensis, but also the impact of climate change and ocean acidification on marine symbiosis. The Roscofensis worm rejects the microalgae when the water is too acid, a bit like coral reefs, and it's a very good educational tool to explain this to younger students. And it actually received an award of the best educational kit back in 2019. So we also use that kit in various do-it-yourself biology contexts, not only in educational context, but also in workshops. Here, like you have photos of the Slovenian workshops, it's a workshop in Slovenia. The biology of S. Roscofensis makes this marine species a special potential model for the development of the bioregenerative life support system applied to space research. You know, it produces oxygen, it recycles CO2 and waste nitrogen metabolism from the animal by the hosted microalgae. It has regenerative capacities of tissues. If you cut its head, it regenerates, it regrows its brain. And it has, of course, energy autonomy with embedded photosynthesis. So the researchers we started to talk, and Xavier Bailly as well, with a program from the European Space Agency called MELISA, Micro-Ecological Life Support System Alternative, where they study loop system for circular life inside the International Space Station. It was established to gain knowledge on regenerative system aimed to the highest degree of autonomy and consequently to produce food, water, oxygen from mission waste. So here you see images from the test plant in Barcelona, and I have also more photos from there. And here you see the loop system with different compartments, whether you have, you see the recycling the crew waste, producing nutrients and extracting the nitrogen that allows it to grow higher plants and or cyanobacteria, photoautotrophic bacteria such as Arthrospira platensis. Here you see the photo. We were visiting the test plant back in November. So on the right, you see this photobioreactor with cyanobacteria, the Arthrospira platensis, spirulina, for whether food supplement, but as well as oxygen production and for the crew of the space station. Here as well, some like control monitoring with different compartments, and you can monitor all the plant from an integrated system. So we are also working with a French space agency as well on an initiative called Open Space Maker to evaluate the design various preparatory experiments to evaluate the capacity of the Roscofensis to embark a space mission. First test was to plan a 3D clinostat, a random positioning machine that simulates zero gravity condition by losing the sense of up and down. So we want to know the behavior of the Roscofensis in zero gravity, but also working on possibly on acceleration columns, radiation test, everything to candidate for the space mission. And we want to design an artistic orbital experiment parallel to the scientific experiment. So a scientific experiment purpose will be how long can organisms survive in microgravity and how they behave compared to the behavior on the Earth, how do they lay the cocoons and the juveniles, and how they intake the microalgae in this environment, how the development phase of the algae in free living stage as well. And one important thing is the regeneration of muscles and brain in microgravity. So we want to first, of course, develop the scientific experiment if everything goes well, and it's probably going to happen only in many years, and if it happens, but we are aiming at that. We want to, of course, recover the results, but also develop them, deploy them in the device in EVA. So here we have this artistic mission scenario where we want to develop what we call a critical container to display on Earth a fully autonomous life support system of the Roscofensis, as well as, let's say, a long-term speculative timeline of the space mission and the different artistic work we will develop along the way on different phases of the research, experiments, activities. And we hope to create some reading groups, speculative narration, and workshops along the exhibition and along the research. So the first exhibition was back in 2018 in Dublin at the Science Gallery Dublin. Here it's another exhibition in Main d'Oeuvre in Paris back in 2020. Of course, the project is a bit slower down with the last 2 years, but we are still working more on technical aspect in the lab. And we're focusing on different aspects, specifically on the circadidal rhythms of the worm itself, because of course it depends on the tides, so of the moon and on the sun, and it gets different light on sun, whether it is in winter or in summer. So it's algorithmic management to be able to manage on the long term the life cycle of the species. I think I'm going to stop here, but I'm going to possibly show you a small video to close it. as a trailer, let's say, if it wants to start. Yes. And I would be happy to. Okay, so just a trailer of the project to come. Just to add, so we are here and it's a good occasion to share and about this research with people such as Ido and Georg and other friends we meet. And next year we also have an event and we're organizing a symposium and hopefully more. And during the ICEA International Symposium of Electronic Arts, that's going to be in France, in Paris, next May in 2023. So in 1 year, right now, from now on. And on the topic is symbiosis. So hopefully we're going to also go on this topic from ocean to space and back to the ocean as environmental research, but also from micro to macro research. So we are very welcome to join us as well. And I guess we will discuss this more later. So I'll be happy to take questions after Maya. So I hope I was not too long, 30 minutes, even less. It's good. Maya, I stop sharing and share again from.
You go directly.
No, because I was looking at. It's embedded. It's embedded.
It works. Okay.
Yeah, it's hard to continue after this presentation. Yes, goofy. So as Ewen said, I'm a resident of Roscosmoi. So I started this project last year in 2021. And as I said, my background is from food. So especially like through fermentation, I learned that fermentation is an ancestral practice as well. It's something which is older and has been inherited in a closed circle. So inherent knowledge passed by generation from mother to daughter, grandmother to granddaughter as well. And I always was fascinated about how knowledge and codes are working outside of a capitalist systems, that so bartering, exchange society, and codes of how societies are working together, which are beyond this level of surface of capitalist society. And within this project of green open food evolution, I started to take a research on microalgae, but mainly seaweed. And so to say, like how seaweed is a food culture coming from East Asian countries, or as I now found out, mainly people who are living in coastal areas. So it's also eaten in Peru, Ecuador, in Zanzibar, in South Africa. So it really unifies many people and tribes as well. And it has always shown this deeper connectivity we have with the oceans. And as I want to start here, like this is a very famous picture from the 70s. It's Apollo. It was taken from the flight of Apollo 11. Many of the people still know it. It was the first time we depicted the planet Earth, like from the outside. So we there found out we called it blue marble, also noticing that actually the term Earth maybe is wrong when Earth is covered by 80% of water as well. This is a newer picture taken by Landsat satellite from NASA, which is also the blue marble, but in the same time, it also shows tricon. So 3 hurricanes, which are covering about 15% of Earth's surface, to therefore also show like the impact that climate change currently has with its mass extinction, acidification of oceans, desertifications. And it's a picture which I thought was really interesting in 2008, published by Facebook, where they showed the activities of people using Facebook as well, also showing that this global connectivity has been also established with ideas of free internet and free information, and also in this kind of Marshall McLuhan way of seeing the world as a global village as well. So seeing the planet or our habitat from space also changes the way how we appreciate or perceive our planet Earth. And those pictures here, for example, I started to source like NASA pictures, and you find like Sahara dust, and like in spring, for example, in Switzerland, we have the red Sahara dust covering the mountains. But you also can see in the left corner in the bottom, the green-blue algae, so the algae blooms, which are as well part of how our ecosystem is thriving. This picture is from last year. This shows the Australian bushfires and the impact seen from outer space. This one is the Thwaites Glacier. So it's also from last year, Doomsday's Glacier, it was called, when a glacier in the size of 80 miles broke off from North Polar, and also shows how our Earth is destabilized in their poles. And coming back to the species of human, like there is also the question, how are we going to feed habitants of our Earth in 2050, for example. So Food Summit was part last year, where we looked at some of the videos. I thought it was rather critical in the sense that they engaged a lot of indigenous people to promote food and food system in a rather kitschy way. But some people maybe also know that Europe, for example, how we feed, how we produce our vegetables. This is the greenhouse of Europe. It's in Madeira, Spain, and it shows that half of the peninsula is covered by greenhouses, also as a part of the organic vegetable production. We also have the center period irrigation system, which is mainly applied in America, but more and more is covering miles and miles of surfaces of planet Earth. And it's an entirely computerized system of how to manage agriculture in a way of 21st century in the goal of feeding Earth's population. So I just talk a little bit that the Green New Deal, which was established, it started in the 30s and 40s. It was inspired by UdSSR ideas of modernizing the state and the distribution of food and creating sectors of production and chains, which nowadays with this Green New Deal changed a lot of geopolitical and geographical landscape of planet Earth itself. Those people who read books from Yuval Harari, they read that it's not only 8 billion of humans, but 40 billion of chickens, 25 billion of pigs, and about 14 billion of cows who are living on planet Earth, as well as so-called domesticated animals. So here again, you have this idea of like survival of the fittest, which is contradicted by this idea that those animal and plants who are living in mutuality or symbiosis with humans, they also have a chance to grow bigger in masses as well. Algae, therefore, has always been underestimated, especially in the Western context, which now is started to be rediscovered, that we didn't use it as a food source as people do in Japan and Southeast Asia and coastal people, as I mentioned before, but it is regarded as a source of protein, of amino oils, of lots of minerals, and also claimed as a superfood to cleanse. And people and scientists just see that there lies a lot of potential in this biomass as food source as well. We also see pictures of this, which is the Sargassum Ocean and the Sargassum Belt, which also last year actually created a belt between African and South American continent for the first time. Some people look at it as an invasive species as well. So algae, for sure, they have like two sides. In one side, they are praised as the solution for Anthropocene times, but on the other side, they also become a threat in the way how we see it. And there is this research we started to do in the topic of harmful algae blooms, seeing that algae blooms are more and more covering our bodies of waters, not only on the ocean side, but in the binary country, in water reservoirs, and lots of the harmful algae blooms, like for example in Switzerland, last year for the first time, a lake was closed down for bathing because dogs got killed and it's dangerous for small kids. And there for the first time, like in Switzerland, like the country where people think we always have a source of fresh water, people really got attentive and alerted about that there is a new threat coming up. Microalgae can be measured by satellites by measuring light emission and therefore also the chlorophyll. And what they found out is that they always have been microalgae in the ocean, so algae blooms on big scales, so similar to in the scale of the plastic patch, like they have always been there, and that there is a deep connection between the ecosystem of our atmosphere, of planet Earth's atmosphere, and the way how algae are producing oxygen. So now we know this fact that more than 50% of our oxygen is not coming from rainforests, but actually from microalgae. So plankton, and here Pleurococcus marinus, which is called this algae that is producing the most of the oxygen. And here I just wanted to bring in like James Lovelock's Gaia hypothesis, which back in the 70s somehow earned a lot of critics. And I guess first of all, it was because the name of it, Gaia, which rather tended to be more esoteric. But in the second part, it's also because back in the 70s, we didn't have the tools to measure this big scale empiric data and collect them as we nowadays trying together through satellite and satellite monitoring, as well as citizen science. So Gaia theory is quite complex in the way how it's the way how we calculate all the different factors that are providing the empiric data of it. And I think when Lynn Margulis joined James Lovelock's Gaia theory, she was the first person who actually on a biochemical level also could explain on how this chain of nutrient exchange between the different bacteria. So for example, when you look at the Winogrodsky column, there you see that sulfuric or cyanobacteria are feeding sulfic-furic bacteria. So one species waste is one species food as well, and creates this endless circle of exchange of material and goods. And the Gaia theory to see as this macro theory of how an entropic system is functioning could maybe also scale down and see in microcosmoses as well. So this idea of our microbiome and how we as well exchange information through this microscopic living and species. And what also Lynn Margulis said, that she talks a lot about the 5 kingdoms of species. So you have the protists, you have the bacteria, the fungi, the animal, and the plants. And to discover that algae are part of the protist is quite, for me, this idea of a queer taxonomy. So to see that this protist kingdom, somehow it's also a way of like creating a bin where you can stuff everything inside you rather don't understand. And in this tree of life, like algae and the protists haven't had as much attention because they have been so alien towards what we appreciate or understand of what life is. So spirulina, and for example, can exist in milieus where no other existence or species survives. And the microalgae, which are eukaryotic cells and compared to prokaryotes in a very early state of existence, have been defining the pillars of what life is and how life would evolve. As well as cyanobacteria also created the first great mass extinction with the oxygenation of planet Earth, and all the beauty that we see within the structure that really shows that life also follows somehow a harmony and rhythms is what I thought was really fascinating. So I went to, from my studies of the microbiome, I got interested in how we could apply this connectivity that humans have with our environment into a study with microalgae. And therefore there was this one article that I took as a source of research, which talks about the lateral gene transfer that happened between the microbiome of East Asian people and bacteria as well. So they say this lateral gene transfer has happened because of the century-long intake and digestion of algae to make people more capable of taking out the nutrition of these algae. So therefore, to see that humans are only connected by breathing and by microbes of our digestion system, like for example, when you travel or when you migrate, your microbiome is changing as well. I started this as a starting point of going also into the research to those early and ancestral way of how we domesticated algae, or if vice versa, what is domestication, who is the subject and who is the object. And this goal to come here to Japan is really this goal to get more in touch with this ancient practice and go to the ocean and see how these people live as well, and do a bit of this research of how farming in the ocean was and still maybe is. So food practices, because food is so inherited in unwritten codexes of behavior and habits. Gestures have to be shown to inherit to the next generation, and somehow they perceive like deep time connections as well. And algae, who are still this alien species, which are the protists, which talks about the propagation of asexual and a sexual circle. So it needs two kind of circles, and it shows that life is something that we don't know when you for example, talk about bacterial sex and how on a microscopic level you can exchange information as well. And so digging into the archives of how early research was being held as well, like in especially the times of beginning of science, I also discovered that here's some examples how you give names also to plants. So for example, with algae, I realized every country has different names for their plants. And I don't talk about taxonomical names, but the local names, which talks also a lot about how we see this and regard this plant as well. And there in this research, I discovered Kathleen Drew Baker, which was a female scientist as well. And in the 30s and 40s, she was one of the first women in Brittany who received a scholarship at Manchester University to actually conduct her PhD and her doctor. And she researched especially on algae itself, because I guess as a woman, they allowed her to study this because it was not popular enough as the nervous system of a human brain. And so she really dig deep into this research, and actually was the first woman or human who discovered the in vitro reproduction circle of algae. And she published her paper in a times when there was the Second World War happening. And Second World War was also a period where Japan, and mainly like this southern area of Kyushu with Hiroshima and Nagasaki and all the bombings which happened there, it disturbed the ocean floors in such a radical way that nori and algae stopped their reproduction circle for several years. And it was due to this article that Kathleen Drew Baker wrote that an exchange between Western and Eastern science happened. And this exchange actually helped also to re-establish the noris in Japan, so to make them grow again. And as we found out, like Catherine Drew Baker received a memorial. So she kind of became this sacred figure where there is an annual festival held in April. And it's a memorial towards her. And she even received the name Mother of Ocean, which I guess if you receive this in Japan, it's quite a big honor. So this time and this journey and this research, we really want to go to Japan to see this site. And as I said, see the farms and the algae farms. And then, of course, to continue this project, and especially with the filming project, we would love to go visit other places as well. So as I said, this kelp forests and this place where you can go into the littoral zone because the tidal differences are big and the ocean is accessible. We really want to encounter more this culture where humans still have a very close relationship to the oceans themselves. So some pictures from Roscoff here. This is on one side, on the right side, that is Kombu Royal. On the left side, this is Kombu de Bretagne. So different kinds of kombu. You also have wakame, you have the spaghetti del mar, and you as well have the nori here. And yeah, I guess this is an example of Peruvian algae, also the long tail of the algae they eat. Some ways of how algae farming is also hosted, where you see the gametogenesis, the fertilization, the young spores, and how they use only the nets. So for algae farming, for example, you don't need fertilizer, they just grow by themselves. No additional petrochemicals. And there, of course, then to say when, based on this article, that the microbiome is changing, maybe also the speculation, like nowadays we live in 21st century, we have so much science, we have CRISPR-Cas, we have citizen science, we have poetry, we have music, we have vibrances. And so this idea of like how we also could change, if there is a possible through this change could be possible through microbiomes. I think, as I learned, that bacteria can adapt. They reproduce every 20 minutes, but humans need a lifetime circle to evolve as well. Like even if you have shortcuts or so, for us humans, like this stage of evolution still will last 10,000, maybe 20,000, 30,000 years. But maybe there's somehow a speculative way of how we can fasten this process also to somehow incorporate the photosynthesis into our lifestyle. And one question is if it's rather on a physiognomic stage, or if it's more that we adapt the photosynthesis into our society as a help and add of a biologic wisdom that we apply in our life to become finally Homo Photosyntheticus. And so this research now is like 1.5 years, and last year we conducted a workshop and a performance, and here you see a small video.
Outside your mouth. Feel free to express your contentment or discontentment.
Temperature. We let it sit and climb up. I know, right? Up,
It was a big existential crisis, and the oldest of Homo Photosyntheticus did not know what to do.
Full of diversity, too much for forming images of individuality.
Yes, I'm gonna stop here. So I haven't said, like, the performance and the workshop was a collaboration with Adam Saretsky and Annabelle Gedera. So Annabelle, she conducted a somatic workshop, and Adam Saretsky extracted DNA from the algae, and we tasted the algae. I was cooking them, and at the end, the audience was asked as well to write speculative ways of how what the Homo Photosyntheticus is, and so the workshops and the performances are also part of the research. So really, this idea to gather stories of participants as well. And this is exactly a year ago that we had this performance.
Question is more about, there's a question in the chat. It's more about the Tribeca Day in April festival.
It's 14th of April.
14th of April, and it's in Kumamoto.
Yes.
At the Sumiyoshi Nature Park in Uto. Yeah, on 14th of May. 14th of April, sorry, 14th of April. And if you have any other questions, yes.
Thank you so much.
Yeah, great. Thank you.
So George, luckily leaving, and do you have any?
Yeah, I'm sorry I have to leave soon, but it was really great to see the overview of both of your projects, and I can't wait to meet you in person and meet you in Japan and do the workshop. And yeah, I think there are many questions, but I would like to give the questions to the audience first and see what's coming. So are there any questions from the audience? So please write them or just unmute and say your word. いろいろな質問ありますか？日本語も大丈夫です。
Can I ask a question?
Yeah, go ahead.
Yeah, thank you very much today in Iwasaki Labo for coming. And then actually before I said in the Iwasaki Labo, I'm interested in the like taxonomy and in terms of coexistence and as a method, like I think about the genome analysis and so on now, and I'm writing the research proposal. And then I would like to ask a question through the green and sea blue, including seaweed and cooking and cyanobacteria. What do you wanna like convey to people?
Yes, maybe you heard from the
Yeah, it's very
my way of
hard.
sorry. Maybe you heard I'm not really the scientist. I think poetry is something very important, and I'm a big fan of Ursula Le Guin, who says that future can only be claimed by describing it, and to describe future in an alternative way is also to create and play with words, and not only with this square ideas of science and the logic of how our earth is functioning tamed us in a way. So I also feel uncomfortable if it comes to be creative with language as well. But this, I guess it's a thing that we slowly also realize that in this last 25 years, there really has been an ontologic twist that we really crave more logic in our narration and explanation of how world is functioning, and the time of using more poetry is good. I guess it's good. It's not easy. It takes a bit of effort, but it's worthful, and you have freedom to do. Yeah.
Okay, I'm very understand, and yeah, I think so. Actually, it's very difficult about that, not like reasonable thing from other people, but how it's very difficult to express using the words and like mathematics, but you like conducted about the feeling and like hearing and yeah, our sense. So I very like agree with that. How to expression, how to express. Yeah, thank you very much.
A question from Katarina on chat.
The title of the book, the Lynn Margulis book, I suppose. You had two books. One is Symbiotic Planet, and the other one is Microcosmos. Two books, at least it's mentioning the Roscoe form in the introductions, and so for me was quite jumped on me when I knew the Roscoe form. I'm from Roscoe, and I knew that, and then I saw those books mentioning it in the introductions of those two books. Really strike me. And yeah, Microcosmos and Symbiotic Planet. I guess that was the books you asked.
Okay, thank you. So Maria, how far back can the origin of seaweed cultivation can be traced? How old? Do you have any knowledge about the oldest cultivation of seaweed?
No, I don't have. I heard, like, I'm also very unsure. I heard numbers like 300, 700, but also 10,000.
Mm-hmm. So is it in the
it's as old as
which area? Which area?
Areas, yeah. Like I heard 700 in Japan. Some people said 10,000, and I think it's even older because it's the period of hunting and gathering. Fishing is hunting, right?
Yeah.
Fishing is hunting.
Right.
Eating seaweed comes together with fishery. Like you see it, it's like the ocean vegetable.
There's a very astonishing story of it's back 13,000 years ago in Latin America, who was supposed to not be populated by humans at the time. They found fossils of humans with seaweed, and they had a seaweed regime and actually eating many species. 22 species of algae were found in a cave. And there was a question, it completely challenged the datation of the presence of humans in this South Chile, and they realized that actually the migration of humans, because what didn't happen by land, but by following the coast. And those humans, like 30,000 years ago, could continuously feed them, eating seaweed, and quite a sophisticated diet, because they would actually have 22 species of seaweed in the diet. So it's called now the kelp highway. All along
I see.
from Alaska to Chile.
Let's see. Any other questions, comments? Yeah, actually, I had a lot of questions about, for example, the Roscoe-Fences. I don't remember the Latin name.
Exactly, Roscoe-Fences.
but during their visit to the lab, I asked them many questions to them, and please. Go ahead.
Oh, I was raising my hand for when you were finished.
Go ahead.
Okay, so I just had a little question, which is, if we become photosynthetic, will we just become obese on a sunny day or anorexic in the winter? But in particular, for people that are really into food, and I know there's some foodies around the world that love food, if we photosynthesize, what will it do to gourmet food, like really delicious meals? If you're photosynthesizing, your mouth might evolve away, just be used for breathing. No eating, maybe no tongue, no taste, no smell, maybe smell, but no eating, no joy of eating. Is this the future?
Yeah, we had this conversation today with CDO actually, and it's might not be full photosynthetic, but we might just be partially and still have a much more sophisticated diet and nouvelle cuisine that will emerge. Maybe we will not eat so industrial food and maybe go to more fine high cuisine, and as more gourmet, because we will eat less but better. And on the other hand, we will not rely on this industrial green revolution type of food.
I also had a discussion lately with another woman who is the opposite of a foodie. So she hates cooking, she hates to eat, she only eats meat and sweet stuff. And for her, it was cooking and eating is just a waste of time also. So she was totally against or for this idea to become photosynthetic, and yeah, it's a big debate as well. Like having no reason to eat anymore would also change the geographical landscape of planet Earth as well. Could solve a lot of issues that we have nowadays. And I think Mr. Hideo Iwasaki today, he mentioned something very, very, very good, that the great oxygenation, which happened 2.5 billion years pre our time, was a big crisis in the ecosystem of planet. Like most 90% of the species got extinct by that time. So
yeah, and I think actually Hideo Izawaki-san had a question as well now about just before Mr. Zaletsky took the microphone.
Sorry?
You had a question, no, about Roscoe-Fences.
Yeah. Yeah, it's extremely interesting animals or yeah, organisms, and yeah, and so by the way, I for those who don't know me, and, I am a researcher on, I'm studying on cyanobacteria, so relatively familiar with the photosynthesis related story, and it's really a good opportunity to know something about the artistic research on cyanobacteria and algae, and thank you very much for that. Yeah, and well, so for, yeah, and also, and I'm studying on circadian rhythms, and so I'm curious about the rhythmic composition in this animal, and because, you know, and the circadian rhythms can be taken as our internal realization of the outer world, you know, and because the rhythmicity inside the body is correlated with the Earth rotation or tidal cycle. So it means some like a cosmological, how can I say, and the frequency inside the body, and it's still already poetic coincidence, and so and in the symbiotic state, of course, and not only the interaction between the body and the environment, but also the relationship between the two multi-species inside the body is a topic. So it's much more complicated, but very interesting one. So and as Adam's the concern or question related, and we discussed that somehow about dystopian and utopian aspects of Homo Photosyntheticus, and yeah, of course, everything should be changed if we can fully or photosynthesize by ourselves, and at the cellular level, and we should have if it's in the so there are several ways of symbio how can I say to become photosynthetic humans, and our cells can be evolved as a photosynthetic cell, or incorporating the algae or photosynthetic organisms as a, you know, not how can I say, the cellular symbiotic way, but just the coexistence, like our gut microbiota. And so there are several ways, and but you know, the photosynthesis is also the very cellular and biochemical ones. So we should have for the stable symbiosis, like the green worm, and they should have developed very specific the biochemical pathways inside the body, and to maintain the algae working for the synthesis. So, and I'd like to know some biochemical details of that, because usually photosynthesis, it's as Maya said, the emergence of the cyanobacteria in 3.5 billion years ago, the hyperoxidation caused genocide. And it's because the oxygen is very, very reactive and toxic to many organisms. It's also even our cells. Oxidation is a dangerous biochemical reaction. So therefore, we have a lot defense system against the oxidation in our cell. But if we have a photosynthesis inside the cells, it should cause the higher defending system and anti-oxygen system. So probably it would be interesting to how to develop such type of antioxidant system, and in addition to how to incorporate the photosynthetic apparatus into humans at the same time.
Mm-hmm.
Yeah. Anyway.
I think we still, at least my friend Dr. Bailly is working out on this species, and it's still a lot to learn from it. Sorry, still I think researching this all this this symbionts or this holobiont works together, the convoluta algae and the worm, and I guess you should come visit and discuss.
Yeah, I need to learn a lot.
Yeah, maybe who knows? Yeah, and about the great oxygenation. Actually, the history of the species, we don't even know how long they live. The worms don't know how long they can live. It's not been able. If now they can manage the long life cycles in the lab with a good algorithmic of the circadial, circadian, circulunar management of their life cycle, maybe they will be able to actually see how long they can live. But for now, like a few years ago, it's just been for 2 years now, so we're really working on that more precisely. Still a lot to learn, yeah. But maybe I have to say, it's like it's true, we are looking to the question of Homo photosyntheticus as a speculation from Margulis, as a metaphor, as the animal alga and the human alga, so becoming ourselves Homo photosyntheticus. Like, if the animal can do it, why human couldn't do it? But I would like to see it also not only as a transhumanist perspective, like we can improve ourselves. In science fiction, you have like Kim Stanley Robinson, who wrote a short story about it, and he was talking about tattooing ourselves with some photosynthetic, from microalgae tattooing, so we could partially become gaining energy from photosynthesis from tattoos, like transgenic tattoos or something. But I like to see it also as a metaphorical shift from more post-humanist, I would say, because we are also in this time of we have to change our society, of our habits, way of living. And I was saying today also this, maybe we have to go out from Homo sapiens, the cognition perspective, back to some more planetary, macro to micro, the photosyntheticus, origin of life, cyanobacteria. And it's also a change of perspective, more, post-anthropocentric, very cognition oriented. We are so proud to be sapiens, and maybe more symbiotic, more metaphorically as well, like a driver for social change, ecological change. So it deals with energy, food, medicine, many aspects. the change we should aim for. So this also, I mean, also come from the, you know, the Roscoe form come from my village, but it's also, I'm getting older and my perspective on life maybe changed. And I discovered this worm only 10 years ago, but I grew up, I never looked at it before. And it's maybe also meaning how I should look at, you know, how do we change our lives now differently, a different gaze on even my own beach in front of my parents' house. So it's about a matter of like, yeah, out of the Homo sapiens somewhere else, out of the Anthropocene to the Cthulhucene, I don't know, but it's a matter of direction.
Okay, I agree. Okay, thank you. Any other questions? Adam again. What about from Kirie-san or Kirie-sanは質問とかないですか？出られないかな。Okay, and about Anais or Kira, or do you have any comments or questions? Okay, it's about 9 o'clock, and I really enjoyed and it's a very inspiring talk, and yeah, from the very scientific as well as the speculative and the very diverging aspects of related topics, and I hope you enjoy the tour from tomorrow, right? So they are flying to Kyushu area, the southern part of Japan, and
the train. You take the train.
Train. Okay, good.
To see the landscape.
Great. Okay. Chat. Okay. Thank you. And so then when they come back to Tokyo, and they will
Doro-san.
Konnichiwa. So and again okay. The read talk and they have a workshop at BioClub on the 31st Tuesday of this May. Please join them, and I think that the talk event itself will be online again and in a hybrid style, right? And the workshop is a very local one, and if the participants in Tokyo area, please join them. And so there is information on Facebook. So please, I guess on chat, George already wrote some information, so please check that. And Oyama-san, something to say? Okay. Okay.
We meet tomorrow.
Okay. Okay. So thank you very much for very. So any other question or comment from? Okay. So okay. Thank you very much. So okay. So we are
joining us.
Yes.
Yeah, we are closing this conference, and thank you for joining, and have a nice weekend. Okay.
Yeah, you too. Bye bye, everybody.
Bye.
Bye.
Okay.